Hello. Today we're going to talk about just a general overview of the genetics of tuberous sclerosis or TSC and how that might affect you and your family. I'm Dr. Clara Chung. I'm a clinical geneticist with the Tuberous Sclerosis Clinic at Sydney Children's Hospital. So just to begin with, a brief overview of tuberous sclerosis. What is it? So as a condition, it's what we call a multi-system condition. So it affects lots of different organs, and different people will have different combinations of what is affected. And in the general scheme of things, it's a rare condition. So not a lot of people in Australia have it. Depending on what paper you read or what studies they do and how they collate those people, it ranges anywhere from between 1 in 6,000 people to about 1 in 13,000 people. And I did this calculation a few years ago, but that probably roughly equates to about 2,000 people in the whole of Australia who have TSC. And so TSC is a genetic condition, which is why a geneticist talks to you about what it is. And just to sort of understand what it does in the body, we have genes that tell our body to grow. So it goes, yep, grow more, grow more, grow more, and then we have genes that tell our body to stop growing. It's like, that is enough. We don't need any more growth here, and so we grow to a normal size based on a balance between these two groups of genes, the genes that cause TSC are part of this pathway. So there are two genes that cause TSC. They're easily named. They're named TSC1 and TSC2, and they like to live together in this thing called a complex. So that's where the complex comes from in TSC. And so they sort of sit together. And this complex, so the TSC complex, is part of this bigger pathway called the mTOR pathway. Now, this is, of course, a very complicated diagram, and I don't expect anyone to understand what is going on here. So to simplify what that actually means, we have TSC1 and TSC2 as that little complex. And what it is they are like brakes. So they're one of those genes, well, they are two of those genes that are the sort of stopping genes. So they're brakes on this mTOR pathway, which is the growth pathway. And so when you have a genetic change or a mutation, whatever you would like to call it, where one of these two genes don't work, the brakes don't work quite so well, and so therefore you have an overactive mTOR. So mTOR works too much, and that's what causes the growths to happen. And so the growths in TSC can happen in various organs, and the most common ones would be your brain, your kidneys, your heart, and on the skin. Okay, but there are also a few other places that it can happen. The other thing that the overactive mTOR causes is differences in sort of organ neurology, if you'd like, right? So differences in learning, and it leads to the epilepsy of TSC, and it leads to autism and other behavioral differences that people with TSC might experience. The good thing about TSC1 and TSC2 affecting this mTOR pathway is that we actually have drugs that we all have already discovered many years ago that work on this pathway and does the exact job. So they're called mTOR inhibitors, and so as the name might suggest, it inhibits mTOR. So it puts in that brake that the TSC1 and TSC2 should have done. And so if you give mTOR inhibitors, we know it reduces that overactive mTOR, and many studies have shown that it reduces the various growths that can happen with TSC1 and TSC2, and we think it probably has some effect on epilepsy and potentially development as well. So even though TS is a genetic condition, realistically it is diagnosed what we call clinically. So just based on the features that a person has, we have it's like a checklist. We have a checklist of the various different things that can be part of TSC, and if you tick enough boxes, you have the diagnosis, regardless of what your genetics say. Okay, and the checklist is made up of things that are very distinctive to TSC. So it's harder to sort of mistaken them for another condition. And you don't need to remember this, but essentially someone needs to have two major features or 1 major and 2 minor features. And if you look at the list, this is the list in and of itself. Okay, so we have the major features on the left and the minor features on the right there. The list is made of things that are on your skin, in your brain, in your kidneys, in your eyes, and in other places like your heart and your lungs. Okay, and you can have any combination of those as long as you have 2, okay, or more. But what isn't part of the criteria? So there are lots of things that people with TSC have that are significant impacts on their life that are not part of the criteria. So things like the seizures and the developmental delay and the autism, right? But they're not part of the criteria because there are hundreds and hundreds of conditions that could cause these various things. And so even though they have significant impact on people with TSC, they're not on the criteria because it doesn't actually quite distinguish. You can't use them to distinguish between what is TSC and what is another condition that we need to think about. And the other thing to note is that so the features of TSC do come on with age. So if we're looking at a newborn baby, they might not have everything we expect them to have, but that's because they're a newborn baby and they haven't actually developed those features yet, but they may in the future. And so these are 2 graphs that I've lifted off different papers, and they both essentially show the proportion of people over time, so with age, who then have those conditions. So, as soon as we find out about it, then they get added to the graph, and so the line or the colored in area goes up. And in this particular graph on the right here, the darker areas were their pediatric patients, so the ones I knew about as a kid, and the lighter areas are those who were diagnosed as an adult. And of course, some of these features they retrospectively figured out happened a little bit earlier on in age. But the important thing to remember about this is that there are some things that can be diagnosed or seen, before the baby is born. And the most significant of that is the cardiac rhabdomyomas that can happen before birth. And so sometimes people are and more people are being found to have TSC based on that prenatal ultrasound that they have. However, they don't happen very early on in the pregnancy. And so most people have a scan at about 20 weeks of pregnancy, and they often are not there yet because they happen later. So usually you need like a 30-something week ultrasound that people would have gotten for some other reason, and that's when they're often discovered. And so a normal ultrasound, of course, doesn't rule out anything. We also have things like seizures that happened early on and can happen and do affect the majority of kids. And most kids will have seizures by probably about 3 years of age, and more than half of them would have had a seizure before the age of 1. And then we have things that happen much later in life, often the kidney things, and so having a normal kidney ultrasound early on doesn't mean that you won't get kidney the angiomyolipoma or cysts later on. And then we have things that happen in adulthood. So typically the LAM, so the lung condition, happens only in adult females. And so we don't need to look for those until someone becomes an adult. But how do we find out about these people? So we sort of mentioned a little bit already, but when we did a sort of look at the people in our clinic, and this is quite consistent with what other people find in their clinics, right? Probably about half of people come to attention because of the seizures. So no one found anything antenatally, we didn't know about anything, and then the kid starts having a seizure, and then we go and look for all the other things, and then we go, oh, actually you have tuberous sclerosis. More and more people, as we were I was talking about, come to attention because they found that cardiorhabdomyoma in pregnancy. And so we start to find out more and more so we found more and more people before they have any problems before birth. There's a small group that are diagnosed because of the pale spots on the skin from a discerning pediatrician or GP or dermatologist, and then we go looking for everything else, and we find those. And then, the last few we know about because they either have a family history, so they have a parent who has TSC, so we know to look for TSC in the kid, or because they have the kidney angiomyolipomas, or they have lung problems, or one of the other concerns. So the question that everyone loves to ask me is, well, we have two genes, all right, with TSC. Is there a difference between those who have TSC1 and those who have TSC2? Like, can we actually tell the difference? So if you look at a group of people with TSC1 compared to a group of people with TSC2, yes, the studies do like to say that and do show that those with TSC2 might have more severe symptoms. They're more likely to have seizures. They're more likely to have a little bit worse outcome from a learning and development point of view. But it's really hard to then say, oh, well, then because your kid has TSC2, they will be more severely affected. And the reason for that is because the picture doesn't actually really look like this. What the picture actually looks like is if we lined up a group of people with TSC1 compared to TSC2 on this sort of graph of how severe their symptoms are, right? The people with TSC1 probably look a bit like this, and then the people with TSC2 look a little bit like this. And so as a group, once again, there are probably a few more on sort of towards the severe end for TSC2, but by no means is that everybody, right? And so if you're in this group or if you're in this group, yes, what we've just said applies, right? Those with TSC2 are probably a little bit more severe than those with TSC1. But what if you're this person or you're this person? That doesn't apply. So the most important thing, more so than what genetic change you have or which of the two genes it is your gene, right, is everyone with TSC is different. Even if they're in the same family, they have the exact genetic change in the exact same gene, they can be completely different, vastly different to each other. And so there are many other factors that go into it. And so only that individual person with TSC can tell us what they'll be like. And sometimes we can't predict until something happens. And so there are two medical terms that I just always like to mention. So the first is this thing called complete penetrance, and the second is the phrase variable expressivity. And what that actually means, right, is everyone who has TSC has something. Sometimes you need to look really, really hard. There are people out there who don't even know that they have TSC until they have a kid with TSC, and then we go look really hard and we're like, oh, actually, you do have a couple of spots that no one's ever actually paid attention to. Or if we then scan their brain, they actually have something on their brain that just never caused much of a problem. And so they went through life not realizing that they have the condition. And that is that variable expressivity. So what is seen in someone with TSC is very different to what would be seen in somebody else with TSC. And so everyone has something, but that something is different in everybody. Okay. So you can't have a genetics talk without talking about genetic testing. So this is where we actually get to the genetic testing part of the whole talk. So why do we even bother doing genetic testing if back, at the beginning I said we diagnosed TSC just based on the tick box, right? The checklist, the criteria that we use. So there's a few reasons why we actually do genetic testing, even though someone already has a diagnosis of TSC. The first is for that person themselves, right? Sometimes it's just nice to confirm the diagnosis. We just like to dot I's and cross T's and know. And sometimes that person doesn't meet the criteria yet. As I was saying, sometimes little babies, because they're a little baby, hasn't developed everything yet. And so having that genetic diagnosis would help, because then we would know that, yes, they are likely to potentially develop more things, and we need to watch them carefully, because the genetic testing has confirmed that this kid has TSC, even if they don't tick all the boxes. The other thing is it's important for the wider family. As I just mentioned, sometimes parents are unknowingly, also have TSC. And if we then discover that they also have TSC, it's important for them, for their health, and potentially then for the even wider family. And of course, having a genetic diagnosis also helps from a peer support point of view. The TSA will always accept clinical diagnoses as well, but sometimes just having the genetic diagnosis helps access a few other things for the family. And also for families where they're still planning to have more kids after that kid who has TSC, they like to know for family planning purposes, either to know that actually they are at risk of having another kid with TSC and they might want to do something about that, or just for the reassurance that this was a new thing that happened in the kid and it won't happen again. And it restores what we call reproductive confidence. So some families, go through, and because they've had one kid with TSC, they're so worried about having another kid with TSC, they never have another kid, even if they really wanted to. And doing the genetic testing sometimes can help reassure them so that they can continue their family plans as they were planning to do. So what are we testing? So back to our little group, our little complex called the TSC complex. So the TSC1 and TSC2 genes, as I said, like to sit together in this little complex. But in reality, in your chromosomes, they actually don't sit together. So the TSC1 gene lives on chromosome 9 and the TSC2 gene lives on chromosome 16. And so they don't really interact with each other from that point of view. More importantly, the TSC2 gene actually sits right next to a gene called PKD1. And PKD1 is a gene that causes polycystic kidney disease. And so what happens in some people with TSC is that they have a deletion. So a little section that was removed or missing that actually crosses over both TSC2 and PKD1. And those people have both conditions. So they have TSC2 and they have polycystic kidney disease. And that is sometimes known as the contiguous deletion. So contiguous meaning that it's right next to each other. All right. And so those people, of course, need to be looked after even more closely from the kidney point of view, because they have both conditions. So whenever we do genetic testing and whenever someone talks to you about it, you'll hear all these words that probably mean nothing and they are really, really confusing, and you probably look a little bit like this. So in hopes of simplifying genetic testing a little bit, when we have your gene, it's essentially a long string of letters down the bottom here that spell out an instruction. So if you think about your chromosome back in the day, sort of a few slides ago, as like your book, right? And your genes are like your individual sentences of that book. And so when you're doing genetic testing, essentially, it's a little bit like this picture, right? You're essentially doing a spell check. You're looking at it letter by letter by letter by letter, to comparing it to what we know is the normal set of letters, to see if there's a difference between that person who has TSC and the person and what is expected to be the normal. Okay, so in a way, it looks a little bit like this. So we have, genes that can change a single letter, okay, or a few letters, right? We have changes that put an extra full stop way too early in the sentence, and so the sentence no longer makes sense. We have genetic changes where we add in extra little letters and then everything gets shifted and nothing, it's all just, gibberish, but from there on in. And similarly, when you take out like little letters, so if you just accidentally forget to write one letter or a few letters, and then everything else is also gibberish from there on in. And so what actually gets tested for most people, it's a blood test. Okay, so it's usually the easiest thing to do. All the labs are very good at getting DNA out of blood, so it's the most common thing that we use. Okay, sometimes we can use a mouth swab, which gets a bit of the DNA from the inside of your mouth. Some people might get a skin biopsy, because, especially if they have like an angiofibroma or something, where it's quite easy to take that little bit of skin where we know that thing has TSC. And some people end up having surgery for their TSC feature for one reason or another. And if they then take out the TSC lesion, so the TSC little change there, we could test that for the TSC change, and what and how far we go from a genetic testing point of view really depends on what we have found or have not found so far and how hard we need to go looking. And so at the end of the day, we have people who have a clinical diagnosis of, yes, you definitely have TS. And we have some people who go, oh, you tick nearly enough boxes, but not quite. So you have possible TS. And the majority of those people, we will find a genetic change. So we'll call it pathogenic, so a disease-causing variant in either TSC1 or TSC2. And the breakup is usually about 2/3 of people have TSC2 and about 1/3 have TSC1. Okay. Then we have a smaller group of people who have what we call mosaic TSC1 or TSC2, and I'll go into mosaicism a little bit later. Okay. And then we have this final group of people where we've done all the genetic testing that we can, but we don't find anything. And they're called the NMI group, or the known mutations identified group. And this group is getting smaller and smaller with better and better technology. But right now, it's probably about 5 to 10% of people who we test. And they still have a diagnosis of TSC, but we just don't know why, or we haven't found the reason why yet. And we presume that they are so low level of mosaicism that we can't detect it. And I'll go into that in a little bit. In fact, I'll go into that right now. So why can't we find the genetic variant? So why do this group of 5 to 10% exist in the first place? So the first reason is sometimes we're looking in the wrong place. So back in the day, people have gone, oh, well, we can't find it in either TSC1 or TSC2. Maybe there's a third TSC gene out there that we just haven't discovered yet. And so that's why we have this group of people who we don't find genetic change in. And people have looked and looked and looked and looked and looked for this third gene, and they can't find it. And realistically, the more recent research articles have shown that these other people who we can't find anything for do have a change in TSC1 or 2. We just haven't looked hard enough for it yet. Okay. The other reason why we could be looking in the wrong place is when we do genetic testing, where it is a little bit like spell check, but it's like spell check with some areas redacted, right? So it looks a little bit more like this, where there's, black boxes over things, because we can't get to every single bit of the gene just because of the technology and the way the genes are spelt sometimes. And so if you happen to have a genetic change in one of the black boxes where we just can't look at it, right? So the technology just misses it every time, then yeah, it'll come back with nothing because we couldn't get to it. Okay. And sometimes, and the most likely reason, it's because of this thing called mosaicism that I just quickly mentioned before. Okay, so it's a bit like when you have, all your normal cells or your normal things are the white ducks here, and you have one or a few that is in that yellow. It's that yellow one. Okay, the more scientific picture is this one here. Okay, so the reason why that happens, okay, is we all start with one cell. So we all start with one egg, one sperm that come together to make one cell, and that cell to turn from one cell into the millions and billions of cells that make up your body, right, to actual human looking person, that cell needs to copy and copy and copy and copy, and that's what this diagram here is. So it goes 1 into 2 into 4, etc., etc., etc. Okay, and in that process of copying, a little bit like if you go back and think about if you're copying from a board or you're copying something and you're just scribing, sometimes you can make a mistake just in that scribing process. And so sometimes what happens is at some point one of them makes a mistake, and then everything else copies from the mistakes because it thinks that the mistake is right? So everything else from that has that little mistake, but all the other ones still copied the right thing. And so then you end up with this group of cells that have a slightly different genetic makeup than everything else. And that's what mosaicism is. It's when there's 2 or more different sets of genetic makeup. And so then that's what this group here is. So that group of green cells in the middle here has the slightly different genetic makeup compared to everything else. And so when you're looking for mosaicism, right, so when you're doing genetic testing for mosaicism, it could be so low that you can't, it's like finding Wally, right? And so you just can't see it because there's so much there. You don't know what we need to concentrate on or what is just noise and random things. So it gets lost in the noise sometimes when it's too low. And so it's there, we just can't see it, just like when we're looking for Wally. And for those interested, Wally is right here in the middle. So the other way to think about it is, right, so it will be like this, right? So you have this landscape of lots and lots of white ducks, and then you have those random yellow ducts scattered in between, and the random yellow ducts are the ones that are causing the problem. Okay. And so, but when we do genetic testing, we just select a group of them to look at. Okay, so it's like looking like this. Okay, so if you look like this, you're like, oh, well, they're all fine. We can't find anything. And then maybe you need to look in a different place, or maybe look at slightly bigger area, and then you find the random yellow one. It was like, oh, there it is. Okay, that is the genetic change. But what if you don't have 6 yellow ducts? What if your level of mosaicism is so low that there is only 1 yellow duct in all of this? Okay, then if you look here, you can't find anything. If you look a little bit harder in a slightly different place, and you still can't find anything, and you have to zoom out quite a lot and cast the net really, really wide before you can find that 1 yellow duct that you were looking for. Okay, and so that's why sometimes genetic testing just can't find it. Okay, we just can't. It's just not precise enough, or we can't find it because we just didn't look deep enough. And so for those people who are in this group, and there are quite a few people in this no mutations identified group, then there are things that we can do now that we couldn't do a few years ago that might help us look for that genetic change and hopefully identify that genetic change. So the first thing is what we call deep sequencing. It's just like going back to the ducks thing, right? Looking even harder. So instead of looking at 100 ducks, we're going to look at a few thousand of them in hopes that we then find it. Because if you look over and over and over and over and over again, then you're more likely to find it. Okay, and there's now a test that's available in the lab in Sydney, in Randwick, that can do this technology in outside, like in the clinical setting. So yeah, if you're one of these people, it might be worth talking to your local geneticist about whether you can have access to that test. Okay, and sometimes testing a different sample might help. Okay, so that's that whole looking in a different place, and you might just find it because you were looking in the wrong place to begin with, especially because blood is actually a really bad sample to use from the point of view of looking at mosaicism. And so sometimes we might want to take a bit of skin. We might want to take a bit of affected skin because we're much more likely to find it in something that is showing signs of TS, like an angiofibroma or even one of those pale spots. And sometimes, as I was mentioning before, if you've had surgery and they've taken a little bit of tissue out that is known TSC tissue, then testing that might give us the best chance of finding what it is, what the genetic change is. Okay, and so I've talked a lot about genetic testing and everything, and I haven't actually gone into how TSC is inherited. Okay, so for the vast majority, or for the majority of people, so around 2/3, okay, it's a new thing that happened in the person or the kid that was not inherited. It's what we call a de novo change. So that mistake just happened when that egg or the sperm that went to make that kid was being made. And so it's there, but it's not inherited through the family. We tested both parents. Now the parent has the problem. Okay. And about 1/3 is inherited from a parent, whether that parent knows that they have TSC or not. Okay. So as I was mentioning before, there are some people who are so mildly affected that they had no idea that they have TSC. And so my practice, anyway, is that I would always test the parents because I have been surprised before. And so it's always worth testing the parents just to double check and make sure that they're not in this side, because of course it very much changes your chance of having another kid with the same condition. And so what are your chances of having a kid with the same condition? So this TSC is what we call an autosomal dominant condition. So we have 2 copies of every gene in our body, okay? And so that's the same for TSC1 and the same for TSC2. Okay, so for whichever one, so let's say this is TSC1 thing, right? So for someone who has TSC, they have 1 copy that is fine and 1 copy that has a genetic change in it. Okay, and so when you have children, you pass on either this one or you pass on this one, and you have no control over that. It's just a random flip of a coin chance. And so we will presume that your partner has 2 normal copies of that same gene because they don't have TSC, and of course it's a completely different thing if you have kids with another person with TSC. But what would happen is you can either pass on this one and this one, okay, and just to speed it up, the other possibilities are this combination, or this combination. There's usually about 4 different combinations, but what happens is this half, right, who has inherited the change would also have TSC, and this half would not have TSC. So about a 50-50, that's where that 50-50 chance comes from. So 50-50 chance of having a kid with TSC or not, depending on whether you've passed on the one with genetic change or the one that does not have the genetic change. So what can you do about it? Oh, sorry, no. Before we get to what can you do about it, for those who have mosaic TSC, the story is kind of the same but slightly different. So as we were talking about, those who have mosaic TSC have a group of cells that are completely normal and a group of cells that have that genetic change in it. And so they have this sort of, that is sort of described as this splotchy sort of appearance. That's what that is showing. Okay. When you pass on a genetic change, right, your sperm or egg only has 1 cell in it. So you either have an egg or sperm that was part of that group with no genetic change, right, so has no mutations in it, completely fine, or you have an egg or sperm that was part of the group that did have a genetic change in it, and then, you know, that particular group, that particular egg or sperm does have a genetic change or does have a mutation in it. And so when you have a kid, if it's the former one where the egg or sperm had nothing, then that baby will not have TSC, okay? And if that sperm or egg did have that genetic change in it, then that baby will have TSC. But the baby will not be mosaic because that baby would have started off at that very first one with the genetic change. And so everything that is copied from there on out also has the genetic change in it. So when a parent has mosaic TS, the kid, if they have TS, is not mosaic. So back to it. Can you do anything about it? So some people go, yep, that's fine. We have this chance. We're very okay with this chance. We'll just take our chances. Okay. And other people are a little bit more anxious about it and do want to figure out if their kid does have TSC or not. And so the first thing that first option that they have is in an already established pregnancy, they can have a procedure such as the chorionic villus sampling, where they take a little bit of the placenta and they can do the genetic test in that sample, and then you'll find out if that baby has TSC or not, and you can do with that information as you will. The other thing that people can do is go through IVF and have that genetic testing with the IVF process, which it happens before the embryo gets implanted back into the womb, and so you start that pregnancy knowing that that baby doesn't have TSC or does if you've implanted one with the genetic change. The important thing to know about these options is that we can't do either of them unless we know the genetic change in that person. So if you were in that a group of 5 to 10% that we don't find a genetic change in, then unfortunately we're back at square one, and all you can do is take your chances because we don't know what we're looking for, so we can't offer the genetic change, which is why the other reason why sometimes we do look really, really hard for genetic change in anybody, because we want them to have as many options as they can, especially from a reproductive point of view. So to conclude, okay, so the take-home messages from this talk is the 2 TSC genes act like a brake on the growth pathway that is the mTOR pathway. Everyone who has TSC is completely different to the next person who has TSC because you all have a different combination of things, even if you have the exact same genetic change. The chance of passing on TSC is 1 in 2, essentially 50%. And if it is something that is important to you or your family, genetic testing can be done to avoid having an affected baby. And if all else fails, your friendly local geneticist is always happy to talk to you about TSC and the chances of it being inherited and the testing and your family planning. So thank you for listening to the talk, yeah, thank you.